In this part of the tutorial, I'll provide some suggestions for dealing with autofluorescence in your samples. Why do we care about autofluorescence? Autofluorescence is fluorescence that already exists in our cells prior to staining. It can vary depending on the cell type or activation state, meaning there can be some cells with more or less signal in the data, and then these signals can interfere with our measurement in different ways. In this example of mouse lung cells, there is signal in various channels in the unmixed data without any staining, which limits our ability to use those channels for detecting markers. Why does this happen? If there is similarity in the profiles of the cellular autofluorescence and the fluorophore signal being used for unmixing, the autofluorescence can be misassigned as fluorophore signals in the unmixed data. Here we see similarity in the peaks for the normalized autofluorescence in PBMCs and in the signals for BB510, Alexa 532, and BB515. As a result, there is signal even with unstained cells in the AF532 and BB515 channels when unmixed. Extracting the autofluorescence as a separate signature provides a much better match for these autofluorescence signals, redirecting them and reducing the undesired signals in the fluorophore channels in the unmixed data. Autofluorescence extraction, however, can create problems in the data if not done carefully. If done well, it can improve the resolution in the data and allow us to correct for variable autofluorescence signatures in different cell types. I recommend considering autofluorescence from the beginning of your panel design and experimental planning. You can't really remove it, but you can work around it. Extraction is not the only way of dealing with autofluorescence. I'm going to cover briefly two other strategies for how to deal with it using gating and panel design, which can be complementary to autofluorescence extraction. A gating strategy can be an effective way of dealing with autofluorescence, especially if paired with careful panel design. In this example, we can create a path to allow clean gating on CD3 versus CD4 to identify lymphocytes by first excluding CD11B positive cells. Without that exclusion, the CD3 versus CD4 plot is heavily contaminated by uncorrected autofluorescence, primarily from macrophages, as we see by this plot of F480 versus CD3. These cells will be autofluorescent in several channels in the data. If you use an FMO or unstained controls while developing your panel, it's possible to figure out which channels will not suffer from autofluorescent contamination, and that would then allow you to determine which markers to put where for the myeloid cell phenotyping. For instance, we would probably avoid putting markers from phenotyping macrophages on APC or BV421 in this case, because we see high background in those channels from the macrophages. The point is, depending on what you want to accomplish with your panel, it may not be necessary to extract autofluorescence. With panel design, a simple approach is to avoid fluorophores with spectral similarity to your normalized autofluorescence signature or signatures. In the case of mouse splenic macrophages here, we might leave out fluorophores in the middle of the violet, the short end of the blue, and mid-far yellow-green, such as those listed here by name. Leaving these out will reduce misassignment of the autofluorescence signature as our panel fluorophores, and consequently as markers in the unmixed data. The middle band here shows fluorophores that would probably be acceptable to use, while the lower band and the list on the right shows fluorophores that we might want to avoid due to spectral similarity to the autofluorescence signature. To show you what effect this can have, let's walk through this example with human PBMCs. Here we're trying to extract a single autofluorescence signature and to try to incorporate CD14 on BB510 in the panel. Notice the similarity between these two profiles. When unmixed with autofluorescence extraction, this similarity creates unmixing dependent spread in the data, as you see by these characteristics spreading and curving of the negative. By changing the panel design slightly, we can resolve this issue. In this case, switching to CD14 on BB510 to a spectrally distinct per-CP allows for better unmixing of the CD14 from the autofluorescence. In order to design your panel, then, you need to know what the autofluorescence will look like. Here are some examples of autofluorescence signatures taken from a 5-laser Cytec Aurora. With human PBMCs, while monocytes are more autofluorescent than lymphocytes, the signatures are actually very similar in profile and cannot be unmixed adequately. As we see in this example on the right, trying to extract the two simultaneously just creates a bunch of unmixing spread. In mouse, macrophages and eosinophils are often the most autofluorescent cells for immunologists. Macrophage populations may exhibit distinct autofluorescence profiles in different tissues, so do not assume that your autofluorescence from the spleen will resemble that in your tumor or tissue sample. Fixation and permeabilization also frequently alter autofluorescence, both in intensity and signature. Unstained samples used for autofluorescence extraction, therefore, must accurately reflect the fully stained sample, meaning they need to be processed in the same way, same protocol. If you're doing in vitro activation assays, such as restimulation for cytokines, it's important to bear in mind that the activation will likely alter the autofluorescence due to changes in the cell's metabolism. In this example with anti-CD3 stimulation, the T cells have increased their autofluorescence in the UV region compared to the unstimulated or fresh cells. For the next portion of the talk, I'm going to briefly provide some tips on how to work with multiple autofluorescences. The most important thing is to first decide if you have a problem where multiple autofluorescence extraction is likely to help, and that this problem cannot be addressed using the other methods. As Maria and Joanne will cover, the unmixed, unstained sample is very informative here. If this has substantial signal in your fluorophore channels and diverse signal, such as these examples here with liver and lung, you may need to extract additional autofluorescence signatures. When trying to extract small populations of spiky autofluorescence, we need enough cells to be able to robustly identify these populations to extract their spectral signatures associated with them. If you only run a few events, you won't be able to see the populations that will appear when you run a million events in your fully stained sample. On some cytometers, there are these Autofluorescence Explorer pop tools that allow you to manually explore the data and select populations that may be causing problems. Signatures extracted from these populations can then be added to your unmixing matrix. It's absolutely critical when using these tools or other methods for multiple autofluorescence extraction that you check that each additional signature is providing a benefit and is not creating unmixing spread. As Maria and Joanne will cover, this means assessing each combination of autofluorescence signatures in an unmixing and looking over the n by n plots with and without that combination of autofluorescence signatures or in comparison to other combinations. I'm doing some work now with Peter Mage and Laura Farrafont to try to automate this assessment of which autofluorescence signatures should be used. So hopefully going forward, this will be easier. There are other workflows, such as those in the publications listed here, that substitute clustering for the process of manual identification of autofluorescence populations. This approach can be more systematic than trying to search through the various combinations of detectors manually to find the autofluorescence populations. We still have the problem, though, of determining which of these signatures to use. In this example, replicating that workflow, I've isolated a mere 10 clusters from the mouse lung, and each of these has a slightly different autofluorescence signature. Since the permutations rise by 2 to the nth power, this gives us 1,024 possible unmixings to assess. My recommendation is to think carefully about what you're trying to achieve when unmixing the autofluorescence. If you're trying to use your panel to ask a specific question, make sure that the autofluorescence doesn't interfere with answering that question. If it does, then move to extraction. I'm going to walk through this example of a mouse with a very weak GFP reporter, where the goal is to measure the GFP in a highly autofluorescent cell type, brain microglia. What we want to do here is find the microglia on the scatter plot. We can do this based on our prior knowledge of where the cells are, or by using approaches such as backgating. In the case of backgating, that would mean preparing a single stain sample with a microglia marker such as CD11b, gating on the CD11b positive cells, plotting those cells on forward scatter, side scatter to find out where they are, and then mapping that position back onto our scatter plot for the AF identification. If we do instead gating on, say, everything, or a different population that is not microglia, we won't be focusing our extraction on the microglial population and we'll extract a different signature. Extracting this microglial population for our AF signature is going to align our CD11b positive cells the microglial population with the negative for the GFP channel. Extracting a different population probably won't effectively do that. Now you'll notice here that we haven't removed other autofluorescence populations, such as this spike into the GFP channel on CD11B negative cells. However, if what we want to do is measure the GFP on the CD11B positive cells, and that's really the only thing we're trying to accomplish, then extracting additional autofluorescences is not critical. To sum up, here are some suggestions for best practices with autofluorescence controls. Importantly, treat your unstained autofluorescence control the same way as the fully stained samples. Use the same protocol. Record enough events to allow you to identify any populations you wish to use for extracting spectra. If you are working with multiple tissues, you should probably record an unstained sample for each tissue source. If you're working with human donors, with a tissue such as lung, where environmental exposure can alter autofluorescence, you may wish to record unstained samples from each donor. If the immune response, stimulation, or assay has altered the cellular metabolism, you may need examples of each condition. Here are two examples of how we might prepare the controls for autofluorescence extraction, one for mouse and one for human. In both cases, we include an unstained sample of the source that we're focused on analyzing, and this is our control for the autofluorescence. In the human example, that's the tumor cells. In the mouse liver example, that's the liver cells. What we're not going to do is extract autofluorescence from cells that will be removed by gating, such as dead cells or doublets. If we have a different way of removing the autofluorescence with our gating strategy, or we're not interested in analyzing those populations, we don't need to extract that autofluorescence, and extracting that autofluorescence is probably not going to improve your unmixing. I'm going to leave you with a list of publications and resources that I found to be helpful for understanding how to deal with autofluorescence.